RWTH Aachen is one of the leading universities for engineering sciences in the world. The areas of medical science and technology play a particularly important role here. In the research field of tissue engineering, the body's own cells are grown in the laboratory to form functional tissues. Modern 3D printing technologies make it possible to generate spatial tissue constructs, created individually for each patient. These 3D printing technologies, known as bioprinting, are being developed at the Department of Dental Materials and Biomaterial Research at RWTH Aachen University Hospital.
On account of the lack of available donor organs and donor tissue, and the clinical problem of possible rejection responses to implanting foreign tissue, in the field of tissue engineering, research teams worldwide are intensively researching the possibility of creating replacement tissue in the lab. In my research group, we are developing novel bioprinting techniques for this purpose. These 3D printing techniques will make it possible to create patient-specific replacement tissue in the lab in the future.
In bioprinting, endogenous cells are embedded in a carrier fluid. Via printing heads specially designed for the sensitive cells, the cell suspensions are printed drop by drop and then stabilized post-output via a chemophysical process. Using a patented technique, it is also possible to create larger three-dimensional replacement structures. The cell structures then mature into functional replacement tissue when the 3D constructs are placed into special bioreactors. Professor Fischer's research group is conducting intensive research into developing printing technologies suitable for bioprinting, as well as developing robotics and control technology. In addition, his team of scientists is working on synthesizing printing carrier fluids for the cells known as hydrogels. Hydrogels are substances which have a high water content, making it possible to effectively store vital cells in them. A well-known natural hydrogel is collagen, the main component of the matrix surrounding our body cells. The special challenge here is to print hydrogels in 3 dimensions. This is why novel hydrogel mixtures are being especially developed for bioprinting. On the one hand, these are printable in 3D, and on the other, they are chemically and microstructurally coordinated with the embedded cells in order to achieve the desired biological cell behavior. There are 2 possibilities for using 3D printed replacement tissue. On the one hand, the technology aims to implant 3D printed tissue which has been subsequently matured in a bioreactor to replace damaged or diseased tissue. As well as their potential use as implants in patients, however, 3D printed cell constructs can also be used according to the organ-on-a-chip principle. Here, in vitro tissue models are used to solve fundamental biological, medical, and pharmacological questions.
The use of bioprinting to produce replacement tissue as an individual patient implant is still largely at the research and development stage. Printing an entire organ, for example an artificial liver, will probably remain a utopian ideal for some time to come. For less complex tissue structures, however, for example partial skin replacement for burns victims, bioprinting technology could open up new possibilities for clinical treatment before long. Also, the alternative use of bioprinting for in vitro models has now reached a very advanced stage of development and is already being used for basic medical research. Furthermore, organ-on-a-chip models have a great deal of potential as screening platforms for new medication in the pharmaceutical industry.